we are really in a different place now than we were 5 years ago with Prader-Willi syndrome, 5 years ago, 7 years ago, when really there were no clinical trials, new clinical trials going on in PWS. So it's been a wonderful thing for our community to be looking at new potential drugs. We are an attractive community in some ways for a drug company that is looking to develop a drug because we're an orphan indication, which means it's a rare disease, and so the drug company has protections, some protections. If they get an approval, they get an extended patent, they get fast track opportunities at the FDA to get approval. So there are a lot of reasons why PWS is a good investment for our company. There's a downside to PWS as well, though. I mean, it is a rare disorder, so there's not that many people with PWS. And once you start, it has to be a certain age and they have to be a certain weight. The pool gets smaller and smaller. There's some additional risk in the PWS population compared to sort of a normal, healthy, obese population for anti-obesity drugs. So a company has to kind of balance that. The other thing that we have is we have a serious unmet medical need. So there's no, in PWS, there's no saying, okay, if we just teach people about eating healthy food, that, that's going to cure the obesity problem. That's just not possible with someone with PWS, maybe when they're a child, but as they grow and they have opportunities, more and more opportunities to be a little bit more independent and have access to food, it becomes a real problem. So the FDA recognizes that. They recognize that this is a medical need, it's a serious unmet need, and that makes it, I don't want to say easy, because the drug companies still have to show that a drug is safe and effective in our population. But it allows the FDA to have a little bit more flexibility about the parameters around those clinical trials. So we do offer some opportunities that some pharmaceutical companies might not otherwise have. And we're a pretty cohesive community, and we're pretty motivated, and that can, I'm sure at times that can be a little bit of a curse to our pharmaceutical friends, but most of the time I'm sure it's really good because we're very enthusiastic. So these are I believe the current clinical trials that are in various stages in PWS. So we all know about oxytocin. There's been several clinical trials, early stage clinical trials, everything from infants to adults. Those have mostly taken place in academic, so University of Florida has a study. There's an ongoing study actually right now at Eric Hollander in New York City. They are almost done with that study, so I'm going to put out a plug. They need 2 more patients to wrap up that study of oxytocin in PWS. That's a phase 2 study. There's potential there. The studies have been a little bit mixed. There seems to be some promise, but we need to do more work to see when and how much and what's going to be the best use of oxytocin. Carbetocin is another drug. It's similar to oxytocin. It's an oxytocin analog, so it's slightly different. It's a synthetic molecule. And very recently, so Ferring Pharmaceuticals had done a phase 2 study that many of you perhaps were involved in. There were several sites across the country. Vanderbilt did a lot of patients. And that data is not published. The rumor mill has it that it was generally positive data. Rumor mill, it was out in a press release, so I'm allowed to say that, right? So Ferring made the decision that they didn't want to develop it further, and so a new company, Levo Therapeutics, they have the rights to license the next step, and we're very fortunate that Sarah Cotter, who happens to be standing right there, is a mom of a young lady with PWS, and she is CEO of Levo Therapeutics, and she will be trying to usher carbetocin through the next phase of clinical development. We need to be a little bit patient because she needs to, a phase 3 is a pretty big trial. She needs to get the best procedure in place, protocol in place. She needs to develop the sites and needs to develop the capital to bring that forward. But we're really excited that is moving forward, hopefully pretty soon. Today we're going to hear from two companies in just a minute, one about diazoxide, one about setmelanotide, so I'll skip over those. There's also an effort that has originated in Europe, AZP 531 by Alize, and they've just completed a phase 2 clinical trial. So you have to do phase 1, phase 2, phase 3, and then you ask the FDA for approval. So they're at phase 2. They presented data at the IPSO meeting in July, and that looked very encouraging. It's an anti-obesity, anti-hunger drug, so they're getting ready for the next step to try to expand that to phase 3. I think many of you are familiar, of course, with the Zafgen drug, beloranib. So that was a difficult story. So they took that drug all the way through to phase 3. They had 2 unexpected deaths during that trial that looked to be related to blood clotting issues that was associated with the use of the drug. So they suspended that program. So in many ways, it was obviously very tragic for the families. It was difficult for the field. But the remarkable thing was it did seem to show that a drug could impact hyperphagia in PWS. So that data was presented at the IPSO meeting, and it did reduce weight in most people, and it did also reduce hunger. So while it's obviously not the story anybody would like to end up with, it does tell you that hyperphagia in PWS is a druggable target. Something can help, and that's encouraging in that way. There's also CBD oil. The company Incise has not started, but they're going to be looking at CBD oil in PWS. That's in the planning stages. It's actually up on clinicaltrials.gov, although we haven't gotten any, additional information. But keep your eyes out for that, because that's going to be a pretty wide-ranging study. I think the ages are going to be like 6 to 65 or something like that. So it's going to be open to a wide age group. And the thought is that their particular kind of CBD oil is they're looking whether it will reduce hyperphagia and also potentially have an weight loss and behavior impact. So that will be very interesting. And then there's some devices that are being looked at in PWS. So FPWR is supporting a study by Dr. Holland in the UK on vagus nerve stimulation, which has been used in the obesity field a little bit to reduce hunger. So the interesting thing about that study is, so previously Dr. Holland did a study in a few individuals seeing if the VNS would help reduce hunger and improve weight loss. And he didn't show that, but there was a big change. It was a very small study, but there's a big change in behavior. And so they're doing a new study now, really trying to look at that aspect of it, whether this stimulation improves behavior in people with PWS. Just, so we have a lot of temper outbursts in PWS, and if we can sort of smooth that out a little bit, it would be very helpful for our families. I know it would be a good thing at our house. And Dr. Butler is looking at transcranial direct current stimulation, which is just a very mild electrical stimulation of certain parts of the brain that may also improve, or they're looking at it to see if it improves appetite. And that's an ongoing study that's being done in adults with PWS through the Prader-WIlli homes of Oconomowoc. So we'll look forward to the outcomes of those. So that's a lot. I mean, when you consider that, I have to tell you, I don't know, 7 years ago or something, we were talking about clinical trial development, and our board is a fairly aggressive board, and when it comes to trying to advance therapies, and they say, we have to get a bunch of things going. What can we do to help, get clinical trials going? And, we think we should be able to, get or facilitate 5 trials going in the next 3 or 5 years. And I was like, there's no way. I mean, if we get 1 trial going, we're going to be really fortunate. And actually, here we are, 5-7 years later, and we have all of these trials going. So that was an example of, the aggressiveness was really, they were right. And I'm so glad that they were, because we have a lot of things going on. So we'll take a couple of minutes, and I'll introduce you, and we'll have an opportunity for, as I said, 2 of the drugs that are in development to be discussed, and you can ask some questions about that. Our first one will be the drug diazoxide, which is now in a controlled release form. The company is Essentialis, and Neil Cowan is the Chief Scientific Officer of Essentialis, and they have done a phase 2 clinical trial with Virginia Kimonis at University of California, Irvine. I think he'll tell us about that a little bit, and then their plans moving forward. So, Neil?
The answer is actually, you did help pay for this trial that I'll describe. So I don't mean, I wasn't disagreeing with him, and J-Lo had nothing to do with these results. So just as a reminder, Teresa mentioned diazoxide. That's the parent molecule of our drug. It's been approved for about 40 years for the treatment of hypoglycemic, hyperinsulinemic conditions. It's the global standard of care in those indications. So there's about 120,000 patient years of data on it that helps us understand and have confidence in the safety profile of our drug. And for example, in 40 years and 3,600 publications on the drug, there are 0 thrombotic events. So it's a powerful set of data that exists from that. Ours is a once-a-day tablet formulation of the drug that we've had under development for some time. We've conducted now 8 clinical trials with that, and I'll describe the Prader-WIlli results. We do have data from other patient populations as well. Just by way of background, the target of our drug, which is what's called the KATP channel, it's very widely distributed in the body. You find it in central nervous system tissue, you find it in the pancreas, fat cells, muscle tissue, and depending on both the disease and the tissue, you have varying effects. So we all know how important hyperphagia is. Hyperphagia, as you heard this morning, and if you've been at other conferences, you know, hyperphagia is really driven by dysregulation in the hypothalamus, and our drug helps mimic the regulatory mechanisms that can bring that under control, and then over longer-term treatment actually improves the regulation of those neurons that are involved in appetite and energy expenditure. So we can blunt the hyperphagia signal and have a pretty marked effect on hyperphagia, which I'll show. In adipocytes and in other tissues that make or store fat, we reduce fat stores by reducing new fat being deposited and upregulating its use for energy expenditure. In central nervous system tissue, Teresa mentioned and others know, aggressive behaviors occur in some reasonable subset of PWS patients, and that's very likely due, based on published research from this year, due to a particular neurotransmitter called GABA. That is, when it's low, you see these aggressive behaviors. And in the context of low GABA, our drug helps amplify that GABA signal and reduces those aggressive behaviors. And then we know hypotonia and low lean body mass are universal characteristics of Prader-Willi. And in the context of that, our treatment with our drug in muscle tissue may increase muscle mass and reduce hypotonia by reducing atrophy. So just on the hyperphagia, I have 2 slides. One here, which shows that basically once the way this worked is everybody started on drug. They were treated through 10 weeks. They stepped up on their dose every 2 weeks, so each of the points that you see along that curve are points at which they would have seen their drug, the dose increased. You can see that on average there was about 35% improvement through the end of that open label treatment, and then they were randomized either to stay on drug or to go on to placebo. And if they went on placebo, they began to have their hyperphagia regress back towards where they were at baseline. And if they stayed on DCCR, they continued to show the improvement in hyperphagia. I'll mention it here. We have seen, we had patients who have now been on the drug for 6 months, and we show very marked improvements in hyperphagia over a 6-month time frame in those patients. Just a couple more notes. Response rate for hyperphagia was quite high at about 90%. At the highest dose tested, some patients didn't end up at the highest dose. We had a subgroup that did. There was more than a 40% reduction. And the magnitude of that reduction using the scale that we measure this on was in line with beloranib in the BEST PWS study, so we can have a significant effect on hyperphagia as was seen in that study in a larger study, or we could anticipate that we could, let's put it that way. And we had patients who were skipping meals and declining food offered to them even after an overnight fast and stopped asking about meals, which are all behaviors that you wouldn't see in the absence of an intervention that was effective. We looked at body composition in the study, and so we looked, this is a procedure called DEXA. We looked at body fat, we looked at lean body mass, and then we looked at the ratio of lean body mass to body fat. And we showed over the course of 10 weeks of treatment about a 4% reduction in body fat, about more than a 5% increase in their lean body mass, and almost a 10% change in the lean body mass to body fat ratio. Again, we see those patterns of improvement persist over the course of longer treatment horizons when we've had patients on it for 6 months. All these effects are independent of growth hormone. So about half the patients in the study were on growth hormone, half were not. And the ones on growth hormone tended to be a little bit younger. We did have patients who varied all the way from about 10 on the low end of age range up to about 22. But we saw exactly the same effects in the context of growth hormone treatment as its absence. And so these are additive to what growth hormone does. Again, at the highest dose over the course of 10 weeks, about a 6% reduction in body fat, almost a 9% increase in their lean body mass, which probably is about where it tops out. We don't see that continue to increase over a lot longer time horizons, and almost a 17% change in that lean body mass to body fat ratio over the course of 10 weeks. So these are certainly in line with, if not somewhat better than what you see with growth hormone treatment over the same time horizon. We did look at a set of Prader-Willi associated behaviors, about 23 of them, simply asking are they present or are they absent, not a quantitative scale. They grouped down into 4 sets of behavior. We had an aggressive, threatening, destructive behavior subcategory, self-injurious behavior, which as you might expect is skin picking and nail biting and so on, obsessive compulsive behavior subset, and then just an other category, if you will. The aggressive one had included such behaviors as screaming and yelling, throwing objects, aggressive, violent actions, foul language, threatening others, and destructive behavior. If you look at it on an individual behavior basis, so one individual might have 2 or 3 of these or none, about 73% of the time individual behaviors stopped over the course of 10 weeks of treatment. But even more notably, at baseline, about 70% of the patients in the study displayed 1 or more of those behaviors, and at the end of treatment, only 20% did. And that p-value, the smaller it is, the less likely it is that it occurred by chance, and you're getting down in some pretty rare likelihood that this was just by chance. So again, strong evidence that regressive behaviors can be reduced, and over the course of 6 months, we saw similar reductions in aggressive behaviors. We saw changes in blood lipids. I won't go into a lot of detail on this. This wasn't surprising to us. It's expected from the mode of action of the drug, but it's beneficial in the context of a patient who's otherwise, over the longer term, and especially if they become obese or at risk of cardiovascular disease, this kind of changes are positive. We looked at insulin sensitivity, which most people don't think about, but if you can improve it, certainly you can have insulin sensitivity or insulin resistance that may be central in in the brain. You can have at the level of the muscle tissue and so on, or even adipocytes can become insulin resistant. We used a measure called HOME-IR, and over the course of over 10 weeks of treatment, HOME-IR improved from about 2.8 down to 0.93. So this is about 67% improvement in insulin resistance. And the interesting thing was when we took patients off a drug, meaning we put them on placebo and treated them for 4 weeks, that improvement in insulin sensitivity was unchanged at the end of those 4 weeks. And we have this, and that's likely to be contributing to improvements in hyperphagia, because if you improve the insulin sensitivity centrally in the hypothalamus and those neurons that regulate appetite, you're going to regulate them better, and you're going to see improvements in hyperphagia. So it's a positive thing, and we see hyperphagia improvements persist for some time after they go off of drug, not only in the first part of the study, but at the end of the 6 months, we had emails from patients that said, I wasn't even thinking about food for a whole nother month after I went off a drug. So I've talked about most of this from the longer term, but we saw an average weight loss in these patients of about 7.3%. So this is quite positive. It's probably slightly better than what was seen in best PWS. On average, there was about a 4% improvement at 6 months, 7.3%. Drug was very well tolerated during this phase. And again, we saw, so basically everything that we saw in the context of the study was reconfirmed in the 6-month treatment, and we showed that the longer term it had this kind of benefit. Safety-wise, we really didn't have any new safety findings. I wouldn't expect it. If I have 120,000 patient years of safety data, if I'm seeing something new, it's not going to be a good thing. But it's possible. We didn't see anything new. Nearly all the adverse events were mild to moderate and improved or resolved while treatment continued. It's actually not true. Most of the adverse events in the double-blind phase occurred in the placebo-treated patients. The majority of them did. And many, I went back and looked carefully and found one that had occurred in a treated patient. And many of the adverse events were common medical complications of Prader-WIlli. Now, peripheral edema is a little different because it's a complication of our drug as well. So we have an effect across some of the highest priority unmet needs. And we have longer-term potential for weight loss and some confirmation that longer-term these effects that we saw in a shorter study are stable. So what are we doing next? This is a general description of the study that we're planning at this point. We would run 2 dose levels of our drug versus placebo. It would be at U.S. sites, some of which are identified there, and we're still looking for a couple of other sites. We'll have a minimum of 75 patients. It could be closer to 90. But 75 lets the study be designed to show that there is significance for the effect of the drug on hyperphagia. Males and females, genetically confirmed PWS, will go down as young as 8 in this study. You know, it says overweight and obese. If in the next discussion with FDA we can get away from that and simply have no criteria related to baseline weight or BMI, we'll do that. But they have to have moderate to severe hyperphagia because that's going to be the basis of the label and approval. We will be looking at aggressive behaviors and quality of life and some other parameters as well. 6 months of treatment, 6 months of open label treatment thereafter. We'll cover travel costs to get to the sites, and we anticipate starting about mid-year. Now, if you were at IPSO, I sounded like it would be earlier in the year, but now it's looking like mid-year. So with that, I'm done and happy to answer any questions about it. So as it turns out, we only had 1 UPD patient in the study. We had 1 screen fail that was UPD, whose BMI was too low and their body fat was too low and couldn't get in. But the UPD patient responded in line with what we saw for the deletion. So I'm not worried about that. And I didn't mention, but the majority of the patients in our study were minorities. We had Hispanics and we had Asians that made up a majority of the patients in the study. And so we're not worried that, and as we broaden out the population, patient population, that and we're going to see a different response than we might have seen. That's what we would see in Southern California. I mean, that's sort of, what does Southern California look like? There's a large Hispanic population, there's a large Asian population, and so it matched that.
Hi again, how are you? Thank you for your talk.
You're welcome.
Nice to see you again. And my question is, what are the side effects? Any negative side effects?
Sure. So first, let me say that there's no side effect that we've seen, or it really appears in the literature, that is, they all will resolve if you take them off of drug, right? So there's no permanent adverse effect. The most common, so what we see a transient increase in their glucose, both the fasting glucose and glucose that you might measure after a meal, so postprandial glucose. And we see HbA1c rise incrementally. In the longer-term treatment, HbA1c wasn't elevated. It's right back where they were at baseline, and the glucose returns back to baseline. But it's an adverse event in some patients to see that. If they had peripheral edema when they walked in the door, first study visit, that was often stable, but it might, I mean, we don't know that it wasn't stable in its absence. It might have worsened a little. If it did worsen a little in our studies, it began to go right back to their baseline by the end of it. We did have a couple of cases of peripheral edema that occurred in the context of the study. Those, rather than being stable, like ones who showed it at baseline and didn't improve during the course of the study, we had a couple, if it occurred in the context of the study, it resolved within 7 to 10 days. And so that's, peripheral edema was another relatively common adverse event. Other than that, there really, it begins to, because everybody was on drug for a while, you can't, you own everything that happened. And so, we had a couple of cases of hypersomnia, but hypersomnia is not uncommon in Prader-Willi itself. We had a couple of respiratory infections. And again, respiratory infections are not uncommon in the patient population. So,
Increased anxiety?
If we looked at, I mean, all we have is presence or absence of anxiety, right? So we had a couple of anxiety cases that went from having anxiety at baseline to not, and one or two individuals who appeared to have some anxiety by the end that at baseline didn't. But we don't know enough about their medical history to know if anxiety was there in their medical history or not. It wasn't that complete a medical history.
And second question, is this available in stores, and is it I know you're doing the trial, but it's been around for 30 years. Is this something you could go to your endocrinologist and get
There's
prescribed?
an oral suspension version that's either 3 times a day or twice a day. It's got a long, bitter aftertaste. It's out there. I know some people who have used it in patients that are this age range. Some of them haven't had the same experience that patients in the study did. And we would expect that our form might have a better safety profile than that. And we know how to dose our drug, how to get patients safely dosed, and that isn't necessarily known for the older version of the drug.
Thank you.
Sure. Yeah. Go ahead, please.
I have 2 questions. One is, have you looked at the patients that were in your study, sort of 6, I don't know when it ended, sort of in the intervening months, and how many of them had sort of reverted back to where they were before with weight gain and that kind of thing, or has there been some residual effect and they are still perhaps
Yeah.
eating a bit less? And the second is, what do you think the likelihood is that the FDA will actually allow you to run your next drug trial without the obesity
Yeah.
and
Those are both good questions. So, with regard to the first one, one of the things that was true is when I talked about patients being on at 6 months, they didn't immediately following the study go on to the 6-month open label extension. There was a lag there. So they went off a drug or off a drug for a few months, sometimes as long as 6 months, then went back on. What we saw, and I don't know if it's the effect of the drug or not. I mean, all we know is the data that we have. If they had moderate hyperphagia as they entered the study originally and showed improvement, they never got back to their baseline even 6 months later. If they had more marked hyperphagia, they went right back to their base, not right back. I mean, clearly there's at least a month or more where there isn't that effect. And otherwise, we have communication from some of the patients that said, first month off a drug, I wasn't giving any thought to food, wasn't skin picking. Second month, it began a little harder, and by the third month, I was worried I was going to regain all the weight that I lost, and was regaining weight even though, according to her, takes AP classes in high school, so she's articulate and self-aware and can communicate these things herself, so she writes the emails. She said, you know, I was regaining weight without a change in diet. So there is, you know, there's some evidence that there's at least a lag before you begin to see that regress. How likely is it? I don't know that I'd call it a coin toss or not. You know, well, our going-in position will be the hyperphagia is critical, and you can show patients who have normal BMIs, overweight BMIs, all of whom have hyperphagia. They're just, so the obesity is a reflection of effectiveness of the controls of access to food, and some of it is how severe the hyperphagia is. So it's both the individual and the context of their care. So why would you force a selection of less effective environmental control as an inclusion criteria to the study, if in fact virtually every patient who's in this age range has hyperphagia to a varying degree? So you go in with that position, first of all, and then second thing is the next most important response to our drug, as we've talked about it with people, is reducing those aggressive behaviors. So if hyperphagia on the one hand is the most important and the reduction of the aggressive behavior is the next most important, then why force the obesity inclusion criteria or overweight inclusion criteria on the study if you're excluding a lot of patients who are going to require, you know, at the end of the day, benefit from this? So we'll go in there and people from Vanderbilt will be there, and maybe Jennifer Miller, and maybe Therese will be sitting on our side of the table to articulate that position, and not just us. So it's coin toss. Yeah, Ian had one.
With the historical use of dioxide, you said there wasn't any safety concerns, but the other way around, is there any historical evidence that PE is reduced?
So not in the clinical data, but in animal models where you induce thromboses, you can show that if you've treated with the drug and induce, that the rate of development of the thrombosis is delayed, or you change the rate of development of the thrombosis. And it's not just an effect of diazoxide, it appears to be a class, the KATP channel activator class, because the other one in the class that does exactly the same thing is nicorandil, which Europeans may know, but it's not approved here. So both of those do that, and it's statistically significant reduction in the rate of occurrence or the rate of formation of thrombosis.